Good day, everybody. This is Chris with the Ancient Scholar. I hope this finds you all doing well and staying safe, happy, and healthy. What I'd like to do today is I'd like to talk about some information that has been arising over the past couple of months. And this is, of course, all over the news and social media now. And hopefully I can make some sense of it, or a little better sense of it. And this, of course, is in reference to all of the so-called SARS-CoV-2 variants. Some people use the term strain, and other people use the term variant, and oftentimes the terms are used synonymously. When looking at this, because I do think definitions are important, it doesn't appear that there is a well-established and agreed-upon difference between a viral variant or a viral strain, at least in reference to the SARS-CoV-2 virus. And of course, this is the virus that causes COVID-19 disease. So it is in that sense, or it is in that spirit, that I'll use the term variant and strain synonymously as well. Even though some people might disagree with that, I think in a vernacular sense, it's acceptable to do that. So let's talk about some of these variants, and I'm going to talk about three specific variants and what some of the implications might be in reference to these variants or strains. And the first one I'm going to talk about is one that has been around. We suspect that it has been around since at least September 2020, so going way back into last year. And this was first sequenced. So essentially what happened was we saw that there was a large spike of SARS-CoV-2, or a large spike of COVID-19 occurring in the southeast portion of England. And so what they did was they began sequencing the genome because they thought they did that in essence looking for any changes or any mutations in the genome. Just because there's a spike, that's interesting. Could there be some viral changes that could account or somehow be related to this spike in cases? And so that's what they did, is they sequenced the genome of the patients that were experiencing this spike. And lo and behold, they noticed that there were, in fact, several notable mutations. In this particular strain, there were somewhere in the neighborhood of about 17 different mutations. So they found approximately 17 mutations. And some of those mutations involved the spike protein, or the S protein. And this is important because when talking about the spike protein, most of the vaccines that we have developed or are currently being developed, okay, those vaccines use the spike protein in various ways. Some may use RNA, some may put the RNA or DNA in a viral vector like an adenovirus. But either way, these vaccines code for the spike protein, and then the body mounts an immune response to the spike protein. So the spike protein is, in essence, the antigen. All right. And you can check out some of my prior videos on this. I go into much more detail than I'll go into today. So the concern is, if you have a variant or a strain that has substantial mutations to the spike protein, it is possible that if those mutations are substantial enough, that the immune response that the body mounts following the vaccines will not be capable of neutralizing these markedly different spike proteins. And this is sometimes sometimes referred to as escape or viral escape. And that is to say that the virus is now resistant to the immunity that the vaccination causes. I want to be very clear that that is not the case as I speak. That does not appear to be the case right now, but it is a concern that we're seeing these different strains and variants, that there could be an escape in the future. And indeed, there is a lot of research going on looking at coronaviruses, and our understanding of coronaviruses is evolving. The standard way of that we used to think about this was that the mutation rates were very slow, very low for coronaviruses, and that they were not similar to, say, influenza viruses, where an influenza virus can have marked mutations. They can actually have very large mutations called genetic shifts that can occur, and you can have substantial rearrangements in the hemagglutinin and the neuraminidase, the H's and the N's, at least in influenza A, and you can get a completely different virus every year, or several different viruses every year. Now, the SARS-CoV-2 virus is certainly not mutating that quickly, and in fact, coronaviruses have proteins called exonucleases that are actually relatively good at error correction. And if you look at it, the mutation rate is, I don't know, somewhere around 1 in 1,000 to 1 in 10,000. So that's not a huge amount of mutation, but you need to remember that these viruses are being produced by the millions and millions and millions in somebody who has an infection. And every time, right, every time that RNA from the virus gets read, or fuses and gets dumped into a cell and copied, right, or created, there is a chance of just a random mutation occurring, right? And if you have millions and millions and millions and millions of this, and even if this is only a 1 in 1,000 or a 1 in 10,000 thing, it's a surefire bet that you're going to get mutations. And in fact, a person who is infected with SARS-CoV-2, who has COVID-19, probably has multiple different variants of the virus in their body. Now, most mutations are not going to be associated with anything special, and indeed, many mutations are just an evolutionary dead end, and that the mutation is can cause a dysfunction of the virus. The virus can't dock, or it's not infectious, and then it dies out, right? It's not able to compete. There's a selection pressure, and it can't compete, and it just dies out. However, other mutations might make the virus more infectious or more resistant to the immune system, and then those, of course, are going to be able to compete with the viruses that don't have those helpful mutations. And then the viruses that have those mutations will become the predominant viral species, so to speak, right? So it is concerning when we see these mutations arise from that standpoint, and so it does definitely warrant investigation. Okay, so going back to this, what was going on in England. So this specific variant was sequenced in December of 2020. All right, and it has been given multiple names, and these names tend to designate the different kinds of mutations and genetic sequences involved. And I'm not going to go into extraordinary detail on that, but there is one term that you may see, the CDC, I believe, sometimes uses this, and it's known as VOC 2021-2-01. VOC stands for Variant of Concern. So Variant of Concern 2012-01. However, the common name for this that is more frequently used is another delineation. It means the same thing, and that is B117. So B.1.1.7, and this tends to be the common name that we see used in the literature and in the news as well, right? So it all refers to the same thing. It is that this variant that arose in England in probably around September and was sequenced in December. All right, and like I said, there are about 17 mutations. Okay, and interestingly enough, it appears that these mutations occurred very rapidly, probably in the same person. All right, in the same person, likely somebody who had a protracted or prolonged infection, right? So they had lots of viral replication. Their viral load was probably pretty high, and that viral replication probably occurred for a substantial amount of time, and the mutations just accumulated in subsequent generations of viral replication. All right. So that's one thing that we're seeing, or that we're keeping track of, and this it's not entirely clear to me. A lot of people are very concerned that it does appear that this may be more contagious than the more standardized SARS-CoV-2 variants that we've been dealing with. There is no evidence that the virus, that this particular strain or variant is more deadly. However, it is more deadly in the sense that, or it could be more deadly in the sense that if it is more contagious, more people will get it, and subsequently more people will die. But they won't die because it causes a more severe COVID-19 disease. Rather, more people could potentially die because more people are going to get this particular variant. That makes sense. There is no evidence that the mutations are making the virus resistant to the immune response that we see with the current vaccines, right? So escape does not appear to be a problem with this particular variant. It really is the possible increase in infectivity. And there are some people that are skeptical of how much more contagious this is. There is something called the founder's effect, where you find something and you can kind of misattribute it. There are people, very, very well-versed virologists, such as Vincent Racaniello, who runs the TWIV This Week in Virology podcast. He is one that has been he's been fairly skeptical of how much more contagious this particular variant is, and other people are far less skeptical. Looking at it altogether, I would say that I would be comfortable in saying there's a good chance that this is probably somewhere around 10 to, I don't know, maybe 70% more contagious. And obviously, the different lifestyle choices of people are going to play heavily into this as well. So that's kind of where I would go. I think there is some evidence. I think the evidence is concerning enough that there probably is some efficacy to the people that are saying that this is maybe more contagious. I don't know where I fall, if I'm closer to the 10 or closer to 70%, but I think there's compelling reason to believe this may be more contagious, this may be more problematic. Okay, there are two additional variants that have arisen. One has arisen in South Africa, and it arose approximately around the same time that we've seen this variant from Southeast England. arise. And this is, of course, this is variant B.1.351. So B1351. And furthermore, there is another variant that has more recently been identified. It was actually identified in Japan, but I believe it was actually identified in patients from Brazil, right? So the origin appears to be Brazil, although it was sequenced by people in Japan. So I'm assuming that somebody from Brazil flew to Japan, got sick, got tested in Japan, got sequenced, and the Japanese then identified this, and this is variant B. .1.1.248, so B11248. Now, the interesting thing about these two variants of SARS-CoV-2 is they have a specific mutation, and it is a very concerning or potentially concerning mutation, and that is mutation E484, I believe, 484. So this is not a variant or a strain, but this is a specific mutation in the RNA molecule that codes for a specific portion of a protein. And what we have found, and this is actually through a lab, the information that I'll be covering here, this actually comes from Bloom Lab. I'll put that up there. Out of the Fred Hutchinson Cancer Research Center. So Fred Hutchinson. All right, so Bloom Lab out at Fred Hutchinson has identified this, and you can reference them for the additional information. So this E484 mutation, why is this so concerning? Well, what Bloom Lab did is they took serum from people who have recovered from COVID-19, right? So you take serum, and remember the serum is the plasma of your blood is where you have all the antibodies. And so you have this serum that's rich in convalescent antibodies or immunoglobulins, IgGs. And what they did is they tested to see how well that serum neutralized these specific variants of SARS-CoV-2. And what they found was that there was some decrease in the ability, there's some reduction in the ability for the convalescent serum to neutralize. I want to be very clear, this is not an escape situation. This is not an escape situation, all right? Because the plasma was still able to neutralize, it was just decreased, all right? And then the other thing that I want to emphasize, because the next concern, and this is all over the news, is, vaccines are going to be less effective for these particular strains and other strains and variants that have mutations like E484 or mutations that are similar to E484. Okay, so I want to be very clear that the current vaccines are associated with a very strong immune response. All right, and there are approximately 20 epitopes. Okay, there are approximately 20 epitopes. So I guess a good question is, well, what the heck is an epitope? Well, an epitope is technically, it's known as what's called an antigenic determinant. So basically, it's the part of an antigen where an antibody binds, right? And so in earlier videos, when I've talked about the spike protein, I drew something that looked like this. All right, so that's the spike protein coming out of the virus, and then we'll draw the antibody, and I just drew, the antibody attaching to the spike protein and neutralizing it. That's an incredibly simplified picture of what happens. And so what you actually get little areas on the spike protein where specific kinds of antibodies can bind, right? So that's actually a more accurate representation, and where that area where the specific antibodies bind is called an epitope. And the vaccines have been designed to provide a polyclonal antibody response. So you have what's called a polyclonal. Let me just write that up here. See if my right. So you get multiple different kinds of antibodies that are produced, and so for a true escape to a virus to be completely resistant to the immune response with current vaccines, you need to have mutations of approximately 20 mutations that directly impact these specific areas of the spike protein where the antibodies bind. Um, and that has not happened at this point. However, the evidence does suggest that over time, with the accumulation of mutations, this may very well occur. And in fact, it has occurred with other coronaviruses. Many of the coronaviruses that cause the common cold, we have actually been researching with serum from from people way back in the 80s and 90s. We've stored their serum away, froze it, and we're now taking it out of the freezers, thawing it out, and testing it on the newer variants of these coronaviruses that cause the common cold. And so that it's the same species, for lack of better words, but it's just a genetically different variant. So you're talking 20, 30 years of mutations, and we're finding that the serums, the sera from back then, are generally not able to neutralize the new versions of these coronaviruses, right? And so if it can happen with other coronaviruses, it's certainly possible it can happen with these current coronaviruses, with the SARS-CoV-2. However, it's probably going to take a few years for this to occur, to maybe more than that. So there's a high likelihood that we're going to get a couple years out of these vaccines. There's certainly a possibility that we could have a new variant arise where you have just the right kinds of mutations that have accumulated to create an escape. That certainly is possible, although I think that the current vaccines are probably going to be effective for a couple of years. And the companies have actually come out and said that within about 6 weeks, they can actually, because really with the RNA vaccines, it's simply a matter of altering the RNA code, right? So you insert a new code, or you create a new code that takes these mutations into consideration, and then you just update the vaccine. And they are assuring us that they can do this within about 6 weeks. Now, as far as the Food and Drug Administration, if this were to occur, would they then mandate that the companies do another phase 3 trial with this modified vaccine? Meaning that, you know, this is something that could take several months to roll out as opposed to 6 weeks to roll out. And that's a question that I cannot answer, but it is at least comforting to know that we're able to respond to an escape event if it were to occur. And this also, in my mind, really supports the importance of sequencing and testing, right? Testing is so critically important on multiple levels. First of all, it's important to identify people that have COVID-19 so they can isolate and prevent spread of SARS-CoV-2. And we can sequence that, and we can check for mutations that that suggest resistance is occurring, right? So testing is critical on multiple levels, and unfortunately, we're just not doing a great job of that, at least here in the United States. And I will say that these new strains have been detected in the United States. First, the B117 variant was first detected in Colorado, and since I believe it's in 8 or more states now. And if this strain or this variant is truly more contagious, it stands to reason that in very short order it will become the dominant variant in the United States, because it will be able to outcompete the less dominant variants. If it doesn't, then that's evidence suggesting that perhaps this particular variant is not as contagious as some people think it is. Only time will tell. But the good news is it does appear that the current vaccines should provide immunity to these newly emerging variants. However, it is still very important that we keep testing and we keep sequencing to identify problematic mutations and variants in the future. Okay, everyone, I think I'm going to cut it off here. Sorry things are so chopped up. The iPad is just being completely unreasonable tonight, and it has been very problematic. It is not working well with my pencil, and my the palm rejection isn't working very well. So it has really been a headache to try to put this video together for you all. I apologize if it's a little choppy. All right, I'm going to cut it off here, and I hope you all take care of yourselves and stay safe. Bye-bye.